All right, that brings us to a little section on how viruses regulate translation in virus-infected cells. Now, there are a couple of goals here. The main one is viruses have evolved to shut off or regulate host translational systems so that they can have it to themselves. They don't want cellular mRNAs being translated. They want the ribosomes, the tRNAs, the amino acids, the whole translation apparatus to their own devices. So they regulate translation. Now, the host in response, typically when infected by a virus, will shut down translation as a way of defending. It's kind of a host defense. So we're going to talk about both mechanisms, host shut-off and virus shut-off. Now, all the mechanisms of regulation that have been discovered in virus-infected cells are mainly target initiation, and those are the mechanisms in this table in blue with the blue line next to them. And I know you can't see these, but this table shows each initiation or elongation or termination protein, the virus that's involved, and what the virus does to the target. So we'll see later that eIF4G, for example, is cleaved in many virus-infected cells. So the point of this table is twofold. First, to show you how many different mechanisms there are by which viruses antagonize host translation. So clearly it's an important thing to take over. It's an important host cell process to take over. And secondly, you see most of the known processes affect initiation, but there are a couple for elongation and termination. So I want to look at just a few of these to illustrate them for you to see how some of them work. And the first involves eIF2. Now, you may remember that eIF2 is part of the ternary complex, and this is essential for translation initiation. In fact, when the 60S subunit joins the 40S at the AUG, GTP, which is part of the ternary complex, is hydrolyzed to GDP. Now, the ternary complex, again, consists of the MET initiator tRNA, eIF2, and GTP. So now we have eIF2 after a round of initiation. We have eIF2 bound to a molecule of GDP. And this is not useful for making a new ternary complex. You have to have GTP present, so it has to be recycled off. So that's done by a protein called eIF2b, which is basically a GTP exchange factor. It binds GDP eIF2 and exchanges the GDP for a molecule of GTP. And then we now have eIF2 GTP, which can go combine with a MET tRNA and make a new ternary complex for initiation. This turns out to be a very crucial point of regulation of translation. The eIF2 subunit can be phosphorylated, as you can see here, and we'll talk about what does that in a moment. When eIF2 is phosphorylated, it will still bind eIF2b, the guanine exchange factor, but it binds so tightly that it cannot be released. So now what happens is all these phosphorylated eIF2 molecules soak up the eIF2b, which happens to be present in cells in low quantities. The amount of free eIF2b goes down, and eventually you cannot make eIF2 GTP, so you inhibit translation. So a very simple modification, phosphorylation of eIF2 inhibits translation. All right, so let's see how this works in a virus-infected cell. Cells contain 3 protein kinases that phosphorylate eIF2alpha, a subunit of eIF2. Again, eIF2 is part of the ternary complex. They're called PKR, PERK, and GCN2. We're going to talk about PKR today, but I just want to point out that PERK is a kinase in the ER that responds to misfolded proteins, to stress. So under conditions of stress, which can happen in many ways, virus infection or low nutrients, this kinase phosphorylates eIF2 and shuts down translation because you don't want to make proteins under conditions of stress. And similarly, GCN2 is a kinase that responds to nutrient levels. And when nutrient levels are low, phosphorylates eIF2. But we're going to look at PKR today, which is a defense against virus infection. So when cells are infected by viruses, one of the early defenses is the interferon system. So here we have virus infecting a cell. It is sensed as foreign by the by the cell, in ways that we'll talk about later in another lecture. The cell responds by producing interferon, these purple triangles. The interferon is secreted from the cell and binds receptors on neighboring cells, and that induces the synthesis of mRNAs that encode many, many different proteins which establish an antiviral effect. Those are called interferon-stimulated gene products. One of these interferon-stimulated gene products is PKR, the kinase that phosphorylates eIF2alpha. So this is an antiviral protein because it phosphorylates eIF2 and shuts down protein synthesis, so the virus can no longer replicate. PKR then is induced by interferon as a consequence of virus infection. As it is made, it is inactive. PKR is shown here. It consists of a kinase domain and 2 double-stranded RNA binding domains. In order for it to be activated, it needs to be phosphorylated. And activation means that it's able to phosphorylate eIF2alpha. The way PKR is activated is by phosphorylation, and the phosphorylator is a neighboring PKR molecule. So what happens is PKR binds double-stranded RNA. It does so via these double-stranded RNA binding motifs. And double-stranded RNA, of course, is made in many virus-infected cells, not only RNA viruses, but DNA viruses as well, by divergent transcription, say, from a double-stranded DNA genome. PKR molecules bind double-stranded RNA, and then neighboring PKRs bound to the same RNA phosphorylate each other. So they activate each other, and now these PKRs are able to go and phosphorylate eIF2alpha. So PKR is induced, remember, by interferon. It's activated by virus infection. The activating mean PKR binds double-stranded RNA, phosphorylates itself, and then it can go on to inhibit protein synthesis. The phosphorylation of eIF2 inhibits host translation. It also stimulates apoptosis, and this is basically shutting down the cell so the virus can't replicate. It turns out that this is a very powerful means of repelling virus infection. And if viruses didn't antagonize it, they'd be wiped out. And almost every virus has some means of inactivating the PKR pathway. That's how important it is. So without a means of antagonizing PKR, viruses wouldn't survive. So let's take a look at a couple of mechanisms for doing this. So here is an example from adenovirus. Adenoviruses make double-stranded RNA during their replication, and this potentially could activate PKR by allowing PKR molecules to bind to the RNA and phosphorylate each other. The virus also makes a short double-stranded RNA called VA RNA I. This does not encode any proteins. Rather, what it does, it is very short and therefore can only bind a single molecule of PKR. How clever is that? So now PKR binds it. It can't get phosphorylated because there's no other room for another PKR to bind. And so there's no shutoff of translation. So the virus has effectively inactivated the PKR system. And that's just one way that viruses do it. There are many others. I just want to show you one more because this is also very neat. This is an example from Vaccinia virus. This virus encodes proteins that are pseudosubstrates. That is, they mimic eIF2alpha. And they fool the kinase PKR into phosphorylating them instead of eIF2alpha. So here's the three-dimensional structure of eIF2alpha. And on the right are 2 Vaccinia proteins that are clearly structural mimics of eIF2alpha. And they are mimics of the part of eIF2alpha that gets phosphorylated by PKR. Here's serine 51. That is where PKR puts a phosphate on to inactivate the molecule. So these two proteins, K3L and M156R, they bind PKR, and they're made in big quantities in infected cells, and they basically sop up all the PKR so it can no longer go to act on eIF2alpha. So they're pseudosubstrates. And on this table, I've listed a number of different mechanisms that are active in many virus-infected cells to antagonize this PKR system, essentially. You can see that a lot of different viruses are involved, a lot of viral proteins or RNAs are involved, and the targets are different. Some of them bind double-stranded RNA, some of them bind PKR, and some are pseudosubstrates. So again, this is a very important pathway, and many viruses have evolved mechanisms to get around it.